In 2008, a tiny bone fragment from a Siberian cave produced a genome that matched neither modern humans nor Neanderthals. It revealed an unknown human population. And it was not the only one. These are 10 lost populations ancient DNA brought back into human history. Number 10. The isolated people of the Tarim Basin. At the eastern edge of Central Asia lies the Tarim Basin, a vast landscape enclosed by mountains and dominated by the Taklamakan Desert. Its extreme dryness preserved human bodies with remarkable detail. Hair remained visible. Clothing retained its shape. Even facial features appeared recognizable thousands of years after death. Some individuals were buried inside boat-shaped wooden coffins. They wore woven wool and lived with domesticated animals, dairy products, and crops that connected the region to distant parts of Eurasia. To early researchers, their origin seemed almost obvious. The clothing, livestock traditions, and physical appearance of the mummies appeared to point westward. Many assumed these people were migrants from the Eurasian steppe who had traveled east and settled in the desert. But appearance is not ancestry. When scientists recovered genetic material from 13 early inhabitants of the Tarim Basin, the expected signal of recent Western migration was largely absent. Instead, the individuals formed a highly isolated genetic group. Most of their ancestry was related to ancient hunter-gatherers who had once occupied a much wider part of northern Eurasia during the Ice Age. By the Bronze Age, that ancestry had become rare and was usually mixed with other populations. In the early Tarim people, however, it remained unusually concentrated. They also carried a smaller contribution related to ancient Northeast Asian groups. What they did not carry was just as important. They could not be explained simply as descendants of the Afanasyevo pastoralists who had moved through nearby regions. Nor did they resemble a recently arrived farming population from Central Asia. Their genetic history appeared deeply rooted in the region. This created a striking contradiction. Genetically, the Tarim communities were unusually isolated. Culturally, they were not isolated at all. They kept livestock. They consumed dairy products. They used crops and technologies that had originated far beyond the basin. Their clothes and burial traditions existed within a world of long-distance trade and cultural exchange. The genomes forced researchers to separate two processes that are often treated as one. Culture can spread because people migrate, but it can also spread because neighbors exchange knowledge, tools, food, animals, and ideas. A community does not have to be replaced genetically in order to transform its way of life. The people of the Tarim Basin had participated in a connected world while maintaining a distinctive ancestry. DNA could identify their biological relationships. It could not reveal the language they spoke. It could not prove that they belonged to a later historical group simply because they occupied the same region. And it could not turn genetic ancestry into a cultural identity. That limitation is part of the discovery. The Tarim genomes showed that objects, clothing, and technology may travel farther than populations. But on a tropical island, thousands of kilometers away, ancient DNA revealed the opposite pattern. There, an entire lineage appears to have been displaced so thoroughly that almost none of its distinctive ancestry remains among the people living on the island today. Number 9. The lost Toalean lineage of Wallacea. Between mainland Southeast Asia and the ancient continent of Sahul lies Wallacea, a chain of islands separated by deep water. Even during the Ice Age, when sea levels were much lower, these islands were never joined to Asia or Australia by continuous dry land. Anyone moving through Wallacea had to cross open water. Modern humans reached this region tens of thousands of years ago. Some continued toward New Guinea and Australia. Others remained on the islands where separate communities developed over long periods. Reconstructing their genetic history has been difficult. Heat and moisture quickly destroy ancient DNA. In tropical environments, a skeleton may survive, while nearly all of its genetic material disappears. At Liang Panning, a limestone cave on the Indonesian island of Sulawesi, archaeologists uncovered the burial of a young woman. She had been placed in a flexed position and lived about 7,300 years ago. Her burial was associated with the Toalean archaeological tradition, known for distinctive small stone tools and finely made projectile points. Inside the dense portion of her temporal bone, enough DNA had survived to reconstruct a genome. It became the first ancient human genome recovered from Wallacea. The result did not match any modern population exactly. Much of her ancestry was related to the deep lineage shared by present-day Papuans and indigenous Australians. That relationship made sense. The ancestors of those populations had crossed Wallacea before reaching Sahul. But the woman from Liang Panning did not fit neatly within either group. Her genome represented a previously unknown combination of ancestry. Her lineage appears to have separated around the period when Papuan-related and indigenous Australian-related populations were also diverging. She also carried Denisovan ancestry. This connected her to an even older history of contact between modern humans and archaic populations somewhere in Asia. Another part of her ancestry was related to an ancient Asian lineage that could not be explained by the people living on Sulawesi today. Together, these signals revealed a missing population. The woman belonged to a lineage that had once existed in Wallacea, but was later transformed, absorbed, or replaced. Several thousand years after she lived, farming populations associated with the Austronesian expansion entered the islands. They brought new languages, technologies, crops, and ancestry. Earlier populations may have mixed with them. Some may have survived in smaller communities. Others may have disappeared. But the genetic profile, represented by the Liang Panning woman, is now difficult to find in the region where she was buried. One genome had exposed an entire missing layer of Southeast Asian history, but the result also demanded caution. This was one person. Her DNA could not represent every Toalean community, every island, or every populations that had crossed Wallacea over tens of thousands of years. Her connection to the artists who created Sulawesi's much older cave paintings remains unknown. Ancient DNA can reveal a lineage. It cannot automatically reconstruct an entire society. Still, the discovery changed how the islands were understood. Wallacea was not simply a series of stepping stones used by people traveling towards Australia. It contained populations with their own histories of isolation, adaptation, contact, and replacement. The Toalean-associated woman showed that a lineage could disappear from the place where it once lived. But the same problem appears on a much larger scale in Africa. People buried near the homeland of a major population expansion might seem like obvious ancestors. At Shum Laka, their DNA revealed that they belonged to a different human history. Number 8. The ancient foragers of Shum Laka. In the grasslands and forests of western Cameroon, a rock shelter called Shum Laka preserves evidence of human activity across thousands of years. Its location made it especially important. The wider region has often been connected with the early development and spread of Bantu languages. Beginning several thousand years ago, Bantu-speaking communities expanded through much of sub-Saharan Africa. Over time, their languages, farming traditions, and ancestry spread across enormous distances. Shum Laka lies near one possible starting region for that expansion. Inside the shelter, archaeologists discovered the burials of 4 children. 2 had lived roughly 8,000 years ago. The other 2 were buried around 3,000 years ago. Their location appeared to place them close to the beginning of one of Africa's largest population movements. It was reasonable to ask whether they represented the ancestors of the Bantu-speaking populations who later expanded across the continent. Their genomes told a different story. All 4 children were genetically closest to present-day hunter-gatherer groups from western Central Africa. They did not carry the ancestry profile that later became dominant among most Bantu-speaking populations. The people living near the proposed homeland of the Bantu expansion were not necessarily the population that later expanded. One of the male children carried a particularly deep Y chromosome lineage known as A00. Among the known branches of the human Y chromosome, A00 diverged extremely early. Today, it is found at low frequency, especially in parts of western Cameroon. Finding it in an ancient child from the same broad region showed that this paternal lineage had existed there for thousands of years. But the complete genomes provided the larger lesson. The 4 children represented a population related to ancient Western Central African foragers. Their community was part of a complex population structure that existed before the expansion of farming societies. The finding disrupted a familiar assumption. A major cultural expansion does not always begin with the people who had occupied the region for the longest time. Populations may move into an area shortly before expanding out of it. Farmers may absorb foragers. Foragers may adopt farming. Languages may spread through communities with different ancestries. And people buried close to the center of a later transformation may belong to a population that was eventually displaced. The Shum Laka genomes also highlighted a serious limitation. 4 children from one shelter cannot describe the entire population of Cameroon or Western Central Africa. Africa contains the deepest human genetic diversity on Earth. But warm climates have destroyed much of its ancient DNA. Every surviving genome offers only a narrow view into a much larger history. The evidence suggests that ancient African populations were highly structured. Groups that appear small or geographically restricted today may preserve ancestry related to communities that were once far more widespread. The children of Shum Laka were not simply early versions of the people who later became dominant in the region. They belonged to a population with its own history. That population may have declined, mixed with newcomers, or survived in smaller groups. The available DNA cannot reconstruct every stage, but it clearly shows that living in the same place does not guarantee continuity through time. On the other side of the world, an infant buried in Alaska revealed a similar break in continuity. The child belonged to a branch near the foundation of the settlement of the Americas. Yet that branch no longer exists as an independent population. Number 7. The Ancient Beringians. More than 11,000 years ago, people lived along the Tanana River in what is now central Alaska. At a site called Upward Sun River, archaeologists found the remains of 2 infants buried beneath a residential structure. One of them, known to researchers as USR1, preserved enough DNA for a high-quality genome. The child had lived in a critical region. During the Ice Age, lower sea levels exposed a vast area between northeastern Asia and Alaska. This was Beringia. It was not simply a narrow bridge crossed in a few days. It was a broad landscape of rivers, grasslands, cold steppes, and scattered resources. Human communities could remain there for generations. Most of the ancestry of indigenous peoples of the Americas ultimately traces back to a founding population connected to Northeast Asia. But researchers had long debated when that population formed, where it remained isolated, and how its descendants divided. The genome of USR1 revealed a branch that had not been recognized before. The child was more closely related to indigenous peoples of the Americas than to any population outside the Americas. Yet she fell outside the major northern and southern Native American branches represented by later groups. Researchers called this lineage the Ancient Beringians. Genetic models suggested that Ancient Beringians and other indigenous American populations descended from the same founding population. That founding group had already separated from its East Asian relatives. Later, the Ancient Beringian branch divided from the ancestors of the other major Native American lineages. The split probably occurred during the last glacial maximum, when enormous ice sheets and severe climates restricted movement across the north. This changed the image of the first Americans. They were not one uniform population, moving south together. They had already begun to separate into distinct branches. Some descendants traveled farther into the Americas and contributed to the populations found across the continents. Ancient Beringians remained in the north. Eventually, their distinct ancestry disappeared. Later, populations entered Alaska. Some may have absorbed ancient Beringian descendants. Others may have replaced them in particular regions. The genome of one infant cannot reveal the final event. It only shows that the branch once existed. That is enough to change the story. Ancient Beringians stood close to the beginning of the human settlement of the Americas. But even a founding branch can vanish as a separate population. The discovery also exposed a deeper truth about founders. Founding populations do not remain frozen in time. They separate. They become isolated. They meet other groups. Their descendants adapt to different environments. Some branches grow. Others remain small and eventually disappear. The people who first entered a continent may contribute very differently to the populations that live there thousands of years later. Ancient Beringians did not emerge from an empty northern world. Their history was connected to Siberia. And when scientists examined ancient genomes from northeastern Siberia, they discovered that the region had been transformed by one population replacement after another. The earliest known group in that sequence had been almost completely forgotten. Number 6. The ancient North Siberians. Northeastern Siberia is one of the most demanding environments humans have ever inhabited. Winters are long. Temperatures fall far below freezing. Resources are scattered. Movement can become dangerous for months at a time. Yet people entered the high Arctic more than 30,000 years ago. At the Yana Rhinoceros horn site, far above the Arctic Circle, humans hunted animals, shaped tools, and survived through the Pleistocene. For years, archaeologists knew that people had lived there. They did not know who those people were. The answer emerged from a study of 34 ancient genomes spanning more than 30,000 years of northeastern Siberian history. The data did not reveal one stable population. They revealed several. The earliest sampled individuals belonged to a previously unknown group researchers called the Ancient North Siberians. They were distantly related to ancient Western Eurasian hunter-gatherers. But they were not identical to the later population represented by the Malata child farther south. Nor were they equivalent to the East Asian-related groups who occupied northeastern Siberia later. They formed their own branch. These people survived at extreme northern latitudes as the Ice Age approached its coldest phase. But their ancestry did not remain dominant. A later movement brought people with stronger East Asian-related ancestry into the region. These newcomers mixed with groups related to the earlier inhabitants. The result contributed to a population described as Ancient Paleo-Siberian. That population had important relationships with the ancestors of indigenous peoples of the Americas and with some later peoples of far northeastern Siberia. The genetic landscape changed again during the Holocene. Another expansion of East Asian-related populations produced groups researchers called Neo-Siberians. Many present-day Siberian communities derive substantial ancestry from those later migrations. Across the entire sequence, each major expansion replaced much of the population that had lived there before. Siberia was not simply a road to somewhere else. Calling it a corridor to the Americas can make the region sound empty. The genomes reveal the opposite. Northeastern Siberia was a homeland. People lived there long enough to form distinct populations. They adapted to the environment. They separated from neighboring groups. They mixed with newcomers. And they were replaced. The history was layered. Ancient North Siberians formed the earliest known layer. Ancient Paleo-Siberians emerged from later population mixture. Neo-Siberians reshaped the region again. The names can be confusing. Ancient North Siberians were not exactly the same as the broader ancient North Eurasian ancestry represented later by the Malata child. The populations were related at a deep level, but they occupied different positions in genetic models and were represented by different ancient individuals. These terms are scientific labels. They are not the names the people used for themselves. The larger pattern matters more than the terminology. Human occupation of the far north repeatedly survived environmental change. But survival in the environment did not guarantee long-term genetic continuity. One population disappeared beneath the ancestry of another. The newcomers later experienced replacement themselves. Every population examined so far left physical remains that could be sampled directly. A tooth, a bone, a burial. The next population is different. Scientists have not yet identified a genome that belongs clearly and completely to one of its members. They detected it as a missing explanation inside the genomes of other ancient people. Number 5. The Basal Eurasians. When farming spread into Europe, it transformed settlement, diet, technology, and population history. Ancient DNA showed that many of Europe's first farmers carried ancestry related to people from the ancient Near East. They were genetically different from the hunter-gatherers who had occupied Europe before them. Researchers attempted to reconstruct the ancestry of these early farming populations. Most of the pattern could be explained by known ancient groups. One part could not. The genomes appeared to contain ancestry from a lineage that had separated extremely early from other non-African populations. Researchers called this inferred lineage Basal Eurasian. The name described its position in a genetic model. Basal Eurasians appeared to have separated before the diversification of many of the other non-African lineages represented in the data. They later contributed ancestry to ancient populations of the Near East. Those populations, in turn, contributed to the first farmers who moved into Europe. But no one has uncovered a burial and identified the individual with certainty as a pure Basal Eurasian. There is no agreed skull, no known language, no confirmed archaeological culture that can simply be assigned to them. Their existence was inferred because the ancestry of sampled populations was easier to explain if an additional early lineage had contributed to them. This is what geneticists call a ghost population. Imagine combining several known colors of paint. The final shade should be predictable, but the mixture contains a tone that none of the known colors can produce. One possibility is that the model is wrong. Another is that an additional source once existed. Population geneticists test different combinations of ancestry. They compare patterns of shared mutations. They ask which model best explains the genomes. In the case of Basal Eurasian ancestry, the repeated signal suggested that an early population had contributed to ancient Near Eastern groups. The signal contained another mystery. Ancient populations with more Basal Eurasian-related ancestry often appeared to carry less Neanderthal ancestry. One explanation is that the Basal Eurasian lineage separated before the main episode of interbreeding between Neanderthals and the ancestors of other non-Africans. Another possibility is that later population mixture diluted the Neanderthal contribution. The exact history remains uncertain. The homeland of the Basal Eurasians is also unknown. Southwest Asia is often proposed because their ancestry later appeared in ancient Near Eastern populations. But ancestry can travel far from the place where it first formed. Finding a genetic component in one region does not prove that the original population began there. It is also possible that the term Basal Eurasian hides a more complex structure involving several early populations rather than one isolated group. The uncertainty does not erase the discovery. It defines what can honestly be claimed. Basal Eurasian is an inferred ancestral component. It is not a reconstructed civilization. It is not the name of a known tribe. The population has no confirmed face. but its descendants carried a measurable genetic trace. This changes the way lost populations can be discovered. Archaeology usually begins with a body or an object and asks who left it behind. Genetic modeling can begin with descendants and ask what missing population must have contributed to them. A population can enter human history as an inference before any direct representative is found. But not every lost population is a ghost. Sometimes one ancient individual provides direct evidence for a vanished group whose ancestry later spread across continents. In Siberia, the genome of a child buried near Lake Baikal connected Ice Age Eurasia to the first peoples of the Americas. Number 4. The Ancient North Eurasians. Around 24,000 years ago, during one of the coldest phases of the last Ice Age, a young child was buried at Wallacea in south-central Siberia. Archaeologists recovered the skeleton, along with objects, from a rich Upper Paleolithic culture. The child became known as MA1. When scientists sequenced the genome, they expected it to clarify the ancestry of ancient Siberians. Instead, it connected populations on opposite sides of the northern hemisphere. The mitochondrial DNA belonged to a lineage associated with ancient Western Eurasian hunter-gatherers. The Y chromosome also occupied a position related to lineages, later found in Western Eurasia and among indigenous peoples of the Americas. Across the rest of the genome, MA1 showed a strong relationship to ancient Western Eurasian populations. That was already unexpected. The child had lived deep inside Siberia. But the next relationship was even more important. MA1 was also genetically close to indigenous peoples of the Americas. He did not show the same relationship to East Asian populations. This meant the ancestors of Native Americans could not be described as descendants from one East Asian-related population alone. Before entering the Americas, their ancestors had mixed with a population related to MA1. The original analysis suggested that a substantial part of Native American ancestry came from a group connected to this ancient Siberian lineage. Later research refined the details, but the central conclusion remained. The founding ancestry of indigenous peoples of the Americas included a major contribution from a deeply ancient Northern Eurasian population. Researchers came to describe this ancestry as Ancient North Eurasian. Related ancestry later appeared across a wide region. It contributed to populations in Europe, Central Asia, Siberia, and the Americas. The original population no longer exists in unmixed form. Its ancestry was scattered through later communities. This makes MA1 more than a point on a straight route between Asia and America. The Ancient North Eurasian story is a story of mixture before migration. A population with strong East Asian-related ancestry encountered a population related to MA1. Their descendants later contributed to the founding populations of the Americas. The genome connected two histories that had previously appeared separate. It also warned against projecting modern geography into the Ice Age. A child buried in Siberia carried a strong relationship to Western Eurasian populations. Ancestry that is concentrated in one region today may have existed much farther away in the past. Modern distributions are the result of later migrations and replacements. They do not reveal where ancient populations originally lived. MA1 was not the only individual with this ancestry. A later Siberian genome from Afantova Gora showed a similar genetic pattern. This demonstrated that related populations survived in the region for thousands of years. Even so, the number of directly sampled individuals remains small. Ancient North Eurasian is a broad ancestry label. It does not describe one uniform society. Different communities may have carried related ancestry while speaking different languages and using different technologies. The discovery still provides one of the clearest examples of a population disappearing while its genetic influence continued far beyond its homeland. Ancient North Eurasians vanished as an independent group. Their ancestry reached the Americas, even though the people represented by MA1 never entered the continent themselves. The next discovery brought ancestry closer to the level of individual families. In a cave in Bulgaria, modern humans carried Neanderthal DNA in segments so long that the Neanderthal ancestors had lived only a few generations earlier. Number 3. The Bacho Kiro population. Bacho Kiro Cave lies beneath limestone cliffs in northern Bulgaria. Excavations uncovered stone tools, ornaments, animal remains, teeth, and small fragments of human bone. Some of the human remains were directly dated to between about 45,930 and 42,580 years ago. They belong to some of the earliest modern humans known from late Pleistocene Europe. Their arrival overlapped with the last Neanderthals. One of the largest questions in human evolution concerns what happened when these populations met. Nearly all living people whose ancestry lies primarily outside Africa carry some Neanderthal DNA. Interbreeding clearly occurred. But how many times did it happen? Did contact end after one early event? Or did modern humans continue to mix with Neanderthals as they entered new parts of Eurasia? The genomes from Bacho Kiro provided a direct answer. 3 early individuals carried roughly 3 to 4% Neanderthal ancestry. That was higher than the average found in most later ancient and present-day non-African humans. But the amount of ancestry was not the most revealing detail. The length of the inherited segments showed how recently the mixing had occurred. DNA is passed from parents to children in large chromosome sections. Each generation, recombination breaks those sections into smaller pieces. If Neanderthal ancestry entered a family thousands of years earlier, the inherited segments should be relatively short. The Bacho Kiro individuals carried several long Neanderthal segments. One person had a Neanderthal ancestor fewer than 6 generations earlier. 2 others appear to have had Neanderthal ancestors only several generations before they lived. These were not distant traces inherited only from the ancient admixture event shared by most non-Africans. Neanderthals had entered the recent family histories of these people. The result pointed to repeated contact. Modern humans moving into Europe did not enter a landscape from which Neanderthals had already vanished completely. For a time, both populations occupied overlapping regions. They encountered each other often enough that several individuals from one early modern human population carried recent Neanderthal ancestry. The genomes contained another surprise. The Bacho Kiro people were not most closely related to the later populations that became dominant in Western Europe. Instead, they shared stronger genetic relationships with ancient and present-day populations farther east, including groups connected to East Asia, Siberia, and the Americas. This does not mean they traveled directly from Bulgaria to East Asia. It suggests that they belonged to an early population expansion, whose relatives contributed more strongly to later Eastern populations than to later Western Europeans. Their lineage in Europe did not remain dominant. Later populations entered or expanded. The genetic landscape changed again. Bacho Kiro therefore contained 2 different histories. The first involved direct encounters between Homo sapiens and Neanderthals within recent family memory. The second involved the disappearance of an early modern human population from much of the later European genetic record. Some related ancestry may have survived farther east. Its contribution to later Western Eurasians was limited. This undermines the idea of one successful modern human migration, spreading steadily across Eurasia. Several populations entered Europe. They may have followed different routes. They encountered Neanderthals under different circumstances. Some left descendants. Others did not. At roughly the same time, another modern human population occupied Central Europe. Its members were related across distant archaeological sites. They lived in a very small community. And after their brief appearance, their branch almost completely ended. Number 2. The Zlatý population. In the Czech Republic, a human skull was discovered in a cave system beneath a hill called Zlatý kůň, meaning golden horse. The skull belonged to a woman who lived during the early period when modern humans were entering Europe. Dating the fossil was difficult because conservation materials had contaminated the bone. But genetic evidence showed that she was extremely ancient. More than 200 kilometers away, at Ilsenhöhle in Ranis, Germany, archaeologists recovered fragmentary human remains associated with a stone tool tradition known as the Lincolnbian Ranesian Gersmano-Wishen. For decades, researchers debated who had made those tools. Neanderthals were one possibility. Modern humans were another. Protein analysis and ancient DNA identified the remains as Homo sapiens. Direct dating placed the individuals at approximately 45,000 years ago. Then their genomes revealed something unexpected. The people at Ranis and the Zlatý woman belonged to the same small population. They were distant relatives despite living at sites separated by roughly 230 kilometers. Segments of shared DNA showed that their communities were connected through a narrow network of ancestry. This was not a large, genetically diverse populations spread securely across Europe. The genomes contained long runs of homozygosity, regions where the two inherited copies of DNA were unusually similar. Such patterns can appear when a population is small and people repeatedly find partners within a limited community. Genetic models suggested a recent effective population size of only a few hundred breeding individuals. Over shorter periods, it may have been even smaller. The group had reached Europe, but its demographic position was fragile. Its place in the wider human family was equally important. The Zlatý Ranis population represented the deepest known split from the major lineage that later produced most non-African populations. They had separated early, not long after the ancient Neanderthal admixture event shared by people outside Africa. Their genomes contained Neanderthal ancestry from that common event. But unlike the individuals at Bacho Kiro, they showed no clear evidence of additional recent Neanderthal ancestors after entering Europe. Two modern human populations could therefore live on the same continent at roughly the same time and have very different histories of contact. Bacho Kiro individuals carried recent Neanderthal ancestry. Zlatý Ranis did not. The difference may reflect separate migration routes. It may reflect where they traveled. It may reflect which Neanderthal communities they encountered. Or it may simply be the result of their small population size and limited time in the region. Because these individuals lived close to the period of the shared Neanderthal admixture event, the length of their inherited segments helped researchers estimate when that mixing occurred. The analysis placed the event roughly between 45,000 and 49,000 years ago. But the most important fact about this population is what happened afterward. There is no clear genetic continuity between Zlatý Ranis and the later populations that became dominant in Europe. They were among the earliest modern humans to survive in the cold environments of Central Europe. They made tools. They moved across the landscape. They maintained connections between distant communities. They entered regions where Neanderthals still lived. Then their branch disappeared. The reason remains unknown. Small populations are vulnerable. Disease, low birth rates, isolation, competition, environmental change, and simple demographic chance can push a community toward extinction or absorption. Europe around 45,000 years ago experienced severe climatic instability, but no single explanation has been demonstrated. The honest conclusion is narrower. This early population did not become a major source of later European ancestry. Reaching a continent is not the same as establishing a lasting population. Human history contains migrations that succeeded and migrations that failed. Zlatý Ranis is one of the clearest genetic examples of an early expansion that left almost no identifiable descendants. Yet, these people were still recognizable as Homo sapiens. Their skulls and bones could be identified. Their tools could be excavated. Their genomes could be placed among other modern humans. The final population was more difficult to see. At first, researchers did not possess a complete skull. They did not know its facial structure. They did not know how far its people had traveled. The discovery began with the tiny bone fragment from the opening of this story. DNA did not merely explain the fossil. DNA revealed the population. Number 1. The Denisovans. Denisova Cave opens into the Altai Mountains of southern Siberia. For hundreds of thousands of years, different human populations used the cave or traveled through the surrounding region. Archaeologists recovered stone tools, animal bones, ornaments, and scattered human remains from its sediments. Among those remains was a fragment from the finger bone of a young female. By appearance, it offered almost nothing. It was too small to show the anatomical features normally used to identify an ancient human group. In 2010, researchers reported its mitochondrial DNA. The sequence was unlike that of modern humans. It was also unexpectedly different from known Neanderthal mitochondrial DNA. The bone appeared to belong to an unknown hominin lineage. Mitochondrial DNA records only one narrow line of ancestry, passed from mother to child. To understand the population more fully, researchers sequenced the nuclear genome. That genome changed the human family tree. The individual belonged to a group sharing a common origin with Neanderthals. But the group had followed a distinct evolutionary history. Researchers named them Denisovans, after the cave where the bone was found. The name did not identify a nation or culture. It identified a population through genetic evidence. A large tooth from the same cave carried closely related DNA. But physical remains remained extremely rare. The genome had appeared before the body. The Denisovan sequence also showed that the population had not vanished without descendants. When compared with living people, it revealed a strong relationship to populations in Melanesia. The first major analysis estimated that Denisovan-related ancestry contributed roughly 4 to 6% of the genomes of the Melanesians included in the study. Later research revealed a much more complex pattern. Denisovan-related ancestry is distributed unevenly across Asia and Oceania. Some living populations appear to carry ancestry from more than one Denisovan-related source. This suggests that Denisovans were not one uniform community confined to a single Siberian cave. They probably included several populations spread across a much larger part of Asia. The cave preserved one part of their history. Living genomes preserved others. One inherited Denisovan variant became especially important on the Tibetan plateau. A version of the EPAS1 gene helps the body function in low-oxygen environments. The ancestors of many Tibetans appear to have acquired this variant through admixture with a Denisovan-related population. The people who contributed it disappeared as an independent group. The adaptation survived. But Denisovans had not mixed only with modern humans. Another bone fragment from Denisova Cave, known as Denisova 11, carried a pattern researchers had never observed so directly. Across the genome, one inherited set of chromosomes was Neanderthal-like. The other was Denisovan-like. The individual had a Neanderthal mother and a Denisovan father. She was a first-generation child of two populations that had separated hundreds of thousands of years earlier. Her father also carried traces of older Neanderthal ancestry. The two populations had mixed before. Her mother was more closely related to Neanderthals, who later lived in Western Eurasia, than to an older Neanderthal previously found inside the same Siberian cave. That relationship revealed movement across enormous distances. Neanderthal populations had traveled east. Denisovan populations occupied or revisited the Altai. Their histories crossed inside one individual. Finding a first-generation child among the small number of archaic human remains with usable genomes suggests that such encounters may not have been exceptionally rare whenever the populations met. That does not mean Neanderthals and Denisovans formed one freely mixing group. They remain genetically distinguishable. Long periods of separation allowed each population to develop its own history, but their boundaries were not absolute. The same was true when Denisovans met modern humans. Human evolution was not a series of clean replacements in which one branch vanished before another arrived. Different populations overlapped. They exchanged genes. They separated again. Their descendants carried fragments of those encounters into the future. The Denisovan genome revealed yet another shadow. Comparisons among Denisovan, Neanderthal, and modern human DNA suggested that Denisovans had received ancestry from an even more deeply divergent archaic population. The identity of that group remains uncertain. It may have been related to earlier hominins living in Eurasia. No confirmed genome has yet provided a complete answer. The discovery of Denisovans opened one hidden branch and immediately exposed the possibility of another. Even the geographic range of Denisovans remains under reconstruction. Genetic evidence connects Denisovan-related ancestry to populations far south of Siberia. A jaw from the Tibetan plateau confirmed that a Denisovan-related population had reached high-altitude environments. Other remains, proteins, and genetic fragments continue to expand the known record. Yet scientists still cannot describe one universal Denisovan face. The populations grouped under the Denisovan label may have differed significantly from one region to another. Their anatomy, social organization, and culture cannot be reconstructed from ancestry alone. They may have created technologies already known to archaeology. But assigning individual objects to Denisovans remains difficult, unless human remains or genetic evidence appear in the same context. This uncertainty is not a weakness in the discovery. It is what makes the discovery so important. Before the Denisovan genome, ancient DNA usually helped clarify the relationships of fossils already recognized as human or closely related to humans. With Denisovans, the order was reversed. A population became visible genetically before it became visible anatomically. The finger bone had no identity until its genome created one. The people had no accepted place in human history until inherited mutations revealed their relationships. The population disappeared. Its ancestry did not. Parts of it survived in Melanesia, New Guinea, Australia, Asia, and in biological adaptations carried by living populations. Parts of its history survived in a child with a Neanderthal mother. Other parts remain hidden inside genetic signals that scientists still cannot fully explain. Denisovans occupy number one because their discovery changed the nature of evidence. They demonstrated that the human family contains branches fossils alone might never reveal. And if one unknown population could remain hidden inside a bone fragment for tens of thousands of years, the visible family tree may represent only a fraction of the people who once existed. Final reflection. What does it mean to disappear? The populations in this investigation did not share one fate. The people of the Tarim Basin remained genetically isolated, while adopting technologies and practices from surrounding communities. Their story showed that culture can travel without large-scale population replacement. The woman from Liang Panning carried an ancestry profile later displaced from Sulawesi. Her genome exposed a lost layer of Wallacean history that modern populations alone could not reveal. The children of Shum Laka lived near a region later associated with one of Africa's greatest population expansions. Yet they belonged mainly to ancient Western Central African foragers, not to the population that later became dominant across Bantu-speaking societies. Ancient Beringians stood close to the beginning of the settlement history of the Americas. Their branch was foundational, but it did not survive as an independent population. Ancient North Siberians occupied one of the most difficult environments on Earth before later migrations repeatedly transformed the region. Basal Eurasians remain a genetic ghost. Their existence is inferred from ancestry that cannot be explained without an early and deeply separated lineage contributing to ancient Near Eastern populations. Ancient North Eurasians disappeared as a distinct group. but ancestry related to them spread into Europe, Central Asia, Siberia, and the Americas. The Bacho Kiro people showed that early modern humans repeatedly mixed with Neanderthals. In some cases, the Neanderthal ancestors had lived only a few generations earlier. Zlatý Ranis demonstrated that one of the earliest modern human expansions into Europe could arrive, survive for a time, and then leave almost no genetic continuity. And Denisovans showed that DNA could identify an entire human population before science understood what that population looked like. These are different forms of disappearance. Replacement removes a population from the majority ancestry of a region. Absorption dissolves a population into its neighbors. Migration carries ancestry away from its original homeland. Extinction ends a branch without substantial descendants. And incomplete evidence can make a population appear absent, even when part of its history survives inside genomes that have not yet been studied. The word lost can therefore be misleading. Some populations are lost only as separate groups. Their genetic material continues inside living human beings. Others are lost to archaeology but visible in mathematical models. Some may already be represented by bones stored in museums, waiting for DNA or proteins to reveal their identity. Ancient DNA also changes the shape of the human family tree. A tree suggests branches that divide and remain separate. Human population history behaves more like a network. Branches separate. Some meet again. Genes pass between them. One population contributes to several descendants. Several populations combine to form one later group. People alive today are not the end points of isolated lines. They are mosaics created through repeated encounters. This does not reduce human identity to ancestry percentages. Genomes cannot preserve most spoken words, memories, loyalties, beliefs, or personal relationships. They cannot reveal what the Tarim people called themselves. They cannot tell us what the woman from Lang Paninge thought about her community. They cannot explain how the children of Shum Laka were mourned. DNA reconstructs biological relationships. Archaeology reconstructs lives through objects, food, movement, landscapes, and burial. Neither form of evidence is complete alone. Together, they reveal a past more complex than either could recover separately. The central discovery is not that some populations failed while others succeeded. It is that disappearance is rarely simple. A population may vanish from the map while remaining inside its descendants. It may survive culturally while changing genetically. It may leave no known descendants, but still alter the history of every group it encountered. Ancient DNA has revealed that human history was shaped not only by the populations that survived, but also by the populations that did not. The genomes sequenced so far represent only a fraction of the people who once lived. In frozen ground and cold caves, DNA can survive for enormous periods. In tropical forests, humid soils, and acidic environments, it often disappears quickly. Entire regions remain genetically underrepresented. Thousands of bones have never been tested. Cave sediments contain fragments that have not been identified. Living genomes still carry patterns that current models cannot fully explain. Somewhere, another tooth may already be sitting inside a museum drawer. Another bone fragment may remain beneath the cave floor. Another population may exist only as an unexplained signal inside the DNA of its descendants. A finger bone revealed the Denisovans. A statistical pattern revealed the Basal Eurasians. How many other human populations are still waiting to be found?